In this study, we will test baking soda or meroplankton of Anabas testudinesis, which also known as ikan puyuh. Baking soda or sodium bicarbonate actually is also used as a leavening agent for bake or cook. From one article that I read, which is Cooking Science, state that actually sodium bicarbonate is a mild alkali. They are not like other fruit ingredients which are basically neutral or acidic. It shows that this sodium has a pH that greater than 7. In this lab analysis, we use pH as a parameter to observe fish eggs on baking soda from fertilization stage to hatchings around 24 hours. The observation starts at 7:40 p.m. on 24 April, which is at post-fertilization, and will end until it starts hatching on the next day. So, to ensure that actually the eggs have been fertilized, the transparent eggs will float. Meanwhile, the unfertilized eggs will still at the bottom. Our objective is actually to determine the effects of sodium bicarbonate to the development of Anabas testudinesis during the embryogenesis. So here are the taxonomy of Anabas testudinesis, which refer to the marine species ORG, which is Kingdom Animalia, Phylum Chordata, Class Eptinopterygii, Order Perciformes, Family Anabatidae, and Genus Anabas.
Hi guys! So now I'm going to explain to you guys for the methodology part. So before I actually start into the methodology part, I will actually explain to you guys about the details of our fishes. So basically, we took out the fish eggs from this tank C, which is with a ratio with 2 males and also 1 female. So these are actually all the details that we had, pre-fertilization and also post-fertilization. So for the pre-fertilization, we actually took the time, and it was 9:02 a.m. And then for the post-fertilization, it was about 7:14 a.m. But we took out the fish eggs for around 7:04 p.m. So now, let's move on to the first part, which is the lab preparation. So for the first step, as you can see here, we have 3 petri dish, which is petri dish A, B, and also petri dish C. So petri dish A indicates the control, which fills with dechlorinated water. So why do we use dechlorinated water? Dechlorinated water is used for us to remove all the chlorines and any kinds of contaminations. So basically these contaminations can actually prevent from the fish eggs to develop well. Then afterwards, we use the electronic balance to weigh the baking soda that we want. So we use 2 treatments, which is low concentration and high concentration, which is stated here from 0.2% and also 0.4%. And after we weigh it, we had 0.5 gram for the low concentration and 1.0 for the high concentration of baking soda. And then after that, we had the dilution part. And we use 250 milliliter of volumetric flask, and also we add dechlorinated water until it reaches its calibration mark. So then let's move on to the next analysis, which is the lab analysis. So basically, before we actually put the fish back into the tank, we have injected the oviparin hormone into the fish itself. Basically, normally we actually inject it into their anal, their part of the new anal, and we inject it into it. So basically, this oviparin hormone actually helps to accelerate the development and also the growth rate of the fish eggs. And then after we injected it, we actually place it in a dark surrounding, and we wait for about 5 to 6 hours. And then after that, around 7 a.m., we actually took out the fish eggs, and we actually noticed that there are some eggs that are float and there are some eggs that are sink. So basically, the eggs that are float are the fertilized eggs, and the eggs that are not fertilized, they will sink at the bottom. So basically, all the unfertilized eggs will actually be removed before we actually took out the fertilized eggs. And then we had exposed it for about 24 hours. We actually analyzed it for about 24 hours. And we actually spread it into 3 petri dish, which is control, and also the treatment decrease, which is low concentration, also high concentration. And then we took out 1 drop of each sample from the petri dish and we put it onto the diffraction slide and we observed it under the microscope. So after that, we analyzed and we also observe the changes between their stages until it goes hatch. So basically all this methodology we actually been took out from the journal from Kimmel et al. 1995. Also, I would like to add about why we actually use the fish tank and why do the fish tank have an air pump is because the air pump can actually help the aeration and also help to remove the chlorine as well.
Hi, my name is Shamira and now I would like to explain the results from our data. So first of all, I want to say that overall from our data, the highest level of the development of the egg until the segmentation stage only, which is for treatment, which is for the control stage. So we proceed with the control egg development. The cell starts to develop at the two cells, and they stops to develop until the 25 somite at 21.27 hour of HPF. So we proceed to the next treatment, which is a treatment B. Treatment B, we are using the 0.2% of soda bicarbonate. So the egg starts to develop during the 2 cell on the cleavage period and also stops to develop at the 2 somite stage on 15.92 hour. And we proceed to the treatment C, which is we are using the concentration of 0.4 sodium bicarbonate. And as you can see from the result, the egg starts to develop at the 2 cells on the cleavage period and proceed until the dome stage, where the egg died on 4.72 hours of HPF. This duration is very short. This is because the egg cannot withstand the high basicity, the high amount of basicity of pH. So as you can see from our graph, we can see that we will start with the control first. The egg starts to develop well, but during the actually our egg died during the 15th somite, so we replace the egg with another one using the egg from the same tank as we take from earlier. So we proceed to the concentration of 0.2, the purple one. The egg starts to develop well, but then they die during the 2 somite because they cannot withstand the pH level of the sodium bicarbonate. And for the last one, for treatment C, on the green color, the egg starts to develop well, but then they die during the dome stage, and the shell is burst. This is because they also cannot withstand the high pH level. They only can survive in their optimal range, which is 6 something to 8 point something. So that's all from me for the result parts. So thank you.
For discussion part, based on our result, the egg in treatment B, which is 0.2% of sodium bicarbonate, could survive until 2 somite, while the egg in treatment C, which is 0.4% of sodium bicarbonate, stopped at dome stage. The result shows that Anabas testudinus egg can tolerate with 0.2% of sodium bicarbonate before they stop from developing to another stage. Based on study by Go et al. 2011, hatching time can be faster in alkaline solution compared to neutral and acidic solution, especially under pH 10. When the pH increase, it will induce the development of the egg, which is faster than the egg in control. Unfortunately, the result shows that egg in both treatment either dead or remain at the certain stage. According to Puvaneswari et al. 2009, embryonic stage can be a good indicator for environmental disruption, as they are sensitive to the changes in the environment. The mistake that have been done during the observation, which could affect the development of Anabas testudinous egg. First, we missed the few stages of the egg development. This is due to the carelessness that occurred during the observation. When we transfer the egg into depression slide, the eggs keep on burst for a few times as we put the cover slip on it. Second, we do not turn off the microscope like when we do not observe the egg. This will cause the egg become dehydrated and affect the development of the egg through the heat from the microscope light. This will cause the development of eggs become slower than the usual. Last but not least, the temperature surrounding during the observation may be one of the factors that will affect the development of the eggs. When the temperature becomes cold, it will slow the egg development, while when the temperature is warmer, it will induce the egg development. This can be proved during the observation, which the table located at the back less exposed to the air conditioner compared to the table at front.
So, as the conclusion, the sample in the constant water, that is dechlorinate water, has the most perfect total osmite compared to the treatment B and C. This is due to the dechlorinized water there is a neutral pH that almost 0.4 as stated in the appendices. So for treatment B and C, the pH is above 8 pH, that is unsuitable for the egg to grow. So as stated at the method, the healthy egg, the fertilized egg, will float on the water while the unfertilized egg will sink at the bottom.